This is the second part of the Chapter 6 Histology AV lecture. In the previous lecture, we discussed epithelial tissue and nervous tissue, and now we're moving into connective and muscular tissue. As with the previous lab, this relationship between structure, function, and location is going to be critical, again, not only in this lab, but also in future labs and also in your future career. So as we're going through these tissues, we're going to highlight the importance of these three components for each of these connective tissues and how changing one aspect compromises the overall physiological function of that particular tissue. Now let's get into our connective tissue. Connective tissue consists of widely spaced cells separated by non-living fibers in a large matrix. So we can say that connective tissue has a high extracellular material to low cellular ratio. So these connective tissues are made up mostly of non-living materials, such as collagen fibers, proteins, and this is different from epithelial tissue, which was made up mostly of living material, of living cells, and it had low amounts of collagen, low amounts of those structural proteins. And you can think about this if you look at this image on the right here of a geodesic dome, and there's two individuals climbing on this geodesic dome. So imagine that everything on this geodesic dome, including the people, represent a tissue. And the actual bars of the geodesic dome represent that extracellular material, those collagen fibers, structural proteins. And the individuals climbing on the geodesic dome represent cells. And so you can see that there's more of this ECM, this extracellular material, and relatively fewer amounts of these cells, the people climbing on it. And we're going to talk about two types of connective tissue. We're going to have fibrous connective tissue, and then we're going to have cartilage connective tissue. Now, we're really going to skip over this cartilage in this lab. We're going to come back to it later in the semester, but just do realize that cartilage is a connective tissue. But today we're going to focus mostly on two types of fibrous connective tissue. The first is dense connective tissue, which is made up of dense regular, dense irregular, and elastic connective tissue. And then we're going to have loose connective tissue, which is made up of reticular, areolar, and adipose tissue. Now, for these fibrous connective tissues, we have, again, dense and loose. And with dense connective tissue, this is, as the name suggests, this connective tissue is going to be densely packed. So fibers fill the spaces in between cells. And the way that these fibers are going to be oriented will dictate what type of tissue this is and the structure and the function of it. And so we have dense regular connective tissue, dense irregular, elastic connective tissue. Now this differs from loose connective tissue in that you're going to have more open space between cells in this tissue, hence the name loose connective tissue. And it's going to be those spaces in between the cells are going to be filled with this gel-like substance and other structural proteins. And again, we got three types: reticular, areolar, and adipose. So let's start off with our first dense connective tissue, and this is dense regular connective tissue. Now, right off the bat, we can see that the fibers of this connective tissue are all running in this roughly linear plane, and they're all parallel to each other. The other thing we notice right off the bat is that I don't see many cells here. I don't see any nuclei. Nothing really jumps out at me. And so we're already starting to see that these tissues are going to have a high ECM to low cellular ratio. So the structure of this connective tissue is densely packed parallel collagen fibers. Each of these individual fibers are called collagen fibers, and it's got this wavy appearance to it. We're going to find this tissue at ligaments and tendons, and this tissue is going to hold bones together. That would be for a ligament, and then it's also going to hold muscles to bones, and that's going to be that'll be tendons. And so because we have this densely packed parallel arrangement of these collagen fibers, this tissue is going to be extremely strong in this parallel orientation. It's going to be really strong unidirectionally. And this is going to be critical for ligaments and tendons because that's where all the stress is going to be. It's going to be in this linear plane. That's where all the stress is going to be on bones and muscles. And so it can hold bones and muscles together quite well as long as the stress is in that one unidirectional plane. But as soon as we apply a stress perpendicular, that's when we're going to have tearing of ligaments, tearing of tendons, because it's really not strong in that direction. And this is why soccer players, football players, basketball players injure ligaments at the knee, the ACL, anytime there's a perpendicular stress put on that joint. And it's simply because that ligament, the ACL, cannot withstand stress in that perpendicular plane. Let's compare that tissue to our dense irregular connective tissue. And again, right off the bat, let's just look at these fibers. And this is very different from our dense regular connective tissue in that there's really no obvious orientation of these fibers. You know, it's pretty haphazard. It's kind of randomly, these fibers all randomly arranged. And so the structure is densely packed collagen fibers, kind of in these random directions. And the location is going to be at the dermis and sheaths around cartilage and bone. And the function of this is that it withstands stresses applied in all directions. That's what we're representing with this arrow here. And so we think about our dermis, that's going to be the deeper layers of our skin. If I pull on my dermis, I can twist it anyway, yank it anyway, and when I let go, it goes right back to its normal orientation. And I didn't do any damage to it. This is very, very different from our dense regular connective tissue, right? If we were to do the same thing, if we were to pull on our dense regular connective tissue in any direction, we would likely tear. And so let's discuss this idea of structure, function, and location with these two tissues. An ACL tear is a pretty devastating injury. You can surgically repair it, but you'd rather not have to do that, right? So why not have dense irregular connective tissue at our knees? We would be able to prevent any ACL injury if we had dense regular connective tissue because we could do whatever we want with it, right? Well, that comes with a trade-off, of course. If we had dense irregular connective tissue in our knees at our ACL, we likely wouldn't be able to stand up straight because just the weight of our body would tilt us over. We wouldn't have the same strength in our knee joint if we had dense irregular connective tissue instead of dense regular connective tissue. Likewise, if we switched and had dense regular connective tissue at our skin, anytime someone playfully gave you a pinch, or if you were even just scratching your arm and causing multi-directional stress on your dermis, you would likely injure that connective tissue. So, having the structure of these connective tissues is critical to the function, and the two of those components are going to interact and dictate where we're going to find these two tissue types. Now we've got elastic connective tissue. And the structure of this tissue is that it contains fibroblasts, which are cells within the tissue that are going to produce structural proteins. And these proteins are going to be made up of a specific protein called elastin. And those elastin are going to form elastic fibers. And these fibers are quite obvious. They show up as these dark squiggly lines with the histology. We find this tissue at the lungs, the trachea, arteries, and the aorta. And the function of this tissue is to expand and recoil. That's what these elastic fibers allow. And so if we just think about this tissue specifically at the arteries, this is where we're going to have blood pumping from the heart to the rest of the body. And arteries are going to experience relatively high amounts of pressure as blood has to pass through. And so the elastic tissue is going to allow blood to pump through the arteries without bursting the artery. And so as the blood is pumped through the arteries, the elastic fibers allow it to quickly expand and then recoil. And this is going to be the major function of this elastic tissue. Now we're moving into our first loose connective tissue, and this is reticular tissue. And reticular tissue, you can think of this as a tissue that forms a net. And a nice way of thinking about this is it's kind of like a grapefruit bag or a clementine bag that you would find at the grocery store. And it sort of exemplifies this because the structure is this loose network of reticular fibers and cells. And reticular fibers are going to be kind of these darker, not quite as squiggly as the elastic proteins, but kind of wavy in appearance. And then these organ cells, these are going to be the cells found within this tissue. You find this tissue at the lymph nodes, spleen, the thymus, and the liver. And what this tissue does is it's going to form this supportive framework or stroma around these organs. Next, we have areolar tissue. And the structure of this tissue is this loose arrangement of various fibers, including collagen and elastic and reticular fibers, and these scattered mast cells throughout the tissue. And a nice way to think about this is areolar should make you think about air, and there appears to be a lot of space or a lot of air within this tissue. That's just a helpful way to remember what this looks like, because it is very open in appearance. The location is at the lower layers of epithelial tissue and around many organs. And the function is that it's going to form this framework at epithelial tissue that allows nerves and blood vessels to pass through. And this is how we're going to get nutrients and nerves to these epithelial tissues. We're also going to find this at fascia. This is kind of this sheath, this connective tissue sheath around muscles that holds muscles in place. And then it's also going to connect different types of tissues together. So we've got a lot going on here with this areolar tissue, and I just want to highlight some of these important structures. First, we have mast cells here. These are going to stain really dark on our slides. And this is going to be a living component of this tissue. And this is going to create these structural proteins that we find at this tissue. Next, we've got elastic fibers, which are, again, these are going to be our squiggly fibers just like we saw with elastic connective tissue. And again, this is a fiber that's made up of the protein called elastin. Then over here, we're indicating reticular fibers. These are going to be thin, pink fibers found within areolar tissue. And then finally, we have our collagen fibers, which are also going to be pink, but they're going to be relatively thicker in appearance. Then our final connective tissue is called adipose tissue. And adipose means fat. So this is fatty tissue. The structure of this tissue is large, empty-looking cells with thin margins. And the nucleus is pressed against the cell membrane. And so here we're showing individual cells. These are called adipocytes. And it appears as though they're empty, but they're really filled with fatty tissue. They're filled with adipose. And that's causing the nucleus to get pushed up against the cell membrane. And that's why we've got this nucleus kind of shoved off to the side. This looks very different than simple squamous epithelium, where we had the nucleus right in the middle of the cell. But because this tissue is filled, these individual cells are filled with adipose, that's why the nucleus is squished off to the side. The location is found, you can find this tissue below the skin, breast tissue, and then also surrounding organs. And the function of this tissue is for energy storage, insulation, and cushioning. Now we're moving into skeletal muscle. Skeletal muscle is made up of long, cylindrical, unbranched cells with striations and multiple peripheral nuclei. We call that multinucleate. The location is skeletal muscle, so biceps brachii, quadriceps, pretty much any muscle that you, in order to use it, you have to think about using it. And then the function is voluntary contractions. So again, these are muscles that we're going to use when we think about using them. That's what voluntary means. And we've got two different ways of representing this muscle. On the left is a slide preparation of muscle tissues cut in transverse plane. And you can see that each of these muscle fibers have nuclei, multiple nuclei, but they're kind of pushed to the side. And in this image, you cannot see the striations on the muscle fibers. Now, on this other image on the right, this is a image of muscle tissue prepared in a longitudinal section. So if we were to cut the muscle with the grain, that's how we would get an image like this. And we can see 1 fiber, 2, 3, and then 4 fibers here. And we see that each fiber is in fact multinucleate. It's got multiple nuclei here. And we can clearly see the striations here. Now, the striations are basically, we'll go into more detail on this in a minute, but striations are basically the lines that separate sarcomeres. And sarcomeres are the functional units of muscles that are actually going to shorten with contraction. Just to reiterate how these muscle tissues are going to be prepared, this transverse section is going to look very different from the longitudinal section. And the way that you can think about this is if you took a bunch of pencils and bundled them together, and you looked at them just from the erasers in the transverse plane, you'd get an image that kind of looks like this. And if you looked at the longitudinal section, you can imagine that this would be like looking at the pencils lengthwise like this. Now, it's important that you can only really see the striations in this longitudinal section. You miss them. They don't become as visible in this transverse section. but either view is fair game for a practical quiz. So make sure that in lab you're looking at both of these slide preparations. Now, to understand what exactly striations are, we kind of have to break this muscle down. And a nice way to visualize this is using this image here. And we've got this overall muscle here. And so think of this as like your biceps brachii. And muscles are made up of fascicles. These are what these fibers here are. And fascicles are made up of individual muscle fibers, and these are the actual muscle cells. And within each muscle cell, you have structures called myofibrils. And myofibrils are made up of sarcomeres. And sarcomeres are the functional units of muscles. And this is actually what shortens when muscles contract. And the lines that divide the sarcomeres, those are called Z lines. And this is what the striations, this is where you get these striations. And for our purposes in the lab, within sarcomeres, you have actin and myosin. Those are the proteins that actually cause muscle contractions. And when those proteins interact, the sarcomeres shorten. And so this is how we get muscle contractions. It's these actin and myosin interacting, and it pulls those Z lines or those striations closer together. And this is how you get muscle contraction. Now we've got cardiac muscle. This muscle is striated, okay, but it's involuntary. This is an involuntary muscle. We don't need to think about beating our heart. That would kind of be pretty annoying if we had to think about making sure that we were beating our heart all the time. So the structure is short branched cells with striations and intercalated discs. Intercalated discs are indicated here, and these are gap junctions between cells, between muscles, cardiac muscle cells. And gap junctions are all about moving ions. These are like ion channels, and this is how you get that electricity in the heart to beat the heart. This is unique to the heart, so this is the location is the heart. And the function is to involuntarily contract the heart. And the cells of these muscle tissues are called myocytes. Now, again, I told you that we do have striations here. Again, those are those individual, they divide sarcomeres, or the Z lines within sarcomeres. But unlike skeletal muscle, cardiac muscle is involuntary. And here's an image of actual cardiac muscle tissue. We have our nuclei within our myocytes here. And these intercalated discs are represented here. They're pretty obvious. They're these dark lines here. And this is pretty typical of an image that you might see on a practical quiz. And a good question would be identify this muscle tissue, and then a follow-up question would be identify this feature, like an intercalated disc or a nuclei. And then finally here we've got smooth muscle. Smooth muscle, the structure is short fusiform cells. This is non-striated, and there's only one nucleus within each muscle fiber. And the location is at the GI tract, arteries, uterus, and the pupil. And the function is sustained contractions. And this is involuntary, so we don't have to think about this. And this is all about moving food through the GI tract. We call that peristalsis. Contractions at birth, when your pupil either contracts or expands. This is all smooth muscle. Okay, for your next practical quiz, this is what you should be able to do. And so you should be practicing this in lab before your practical quiz on lab 6. First, you should be able to identify all tissues that we presented in this AV lecture using a microscope. You should be able to describe the structure of these tissues using the techniques that we discussed in the AV lecture, as well as provide a function of the tissue and a location. You should also be comfortable identifying unique features of a tissue, so goblet cells, glial cells, cilia, microvilli, and know what the functions are for each of those unique features. And then finally, you should have a clear understanding of the relationship between structure, function, and location. This is going to be critical not only in this lab, but moving forward in the semester and in your careers. So understand, why does simple columnar epithelium have microvilli? Why does pseudostratified ciliated epithelium have cilia? And what were to happen if those tissues did not have those features, or they were in a different location? This is how you start to have a good understanding of the relationship between structure, function, and location.